So I really have to thank Dr. Simon for making my job very easy. So that was a fantastic introduction of the disease process, and why this is so important. And when I come to work each morning, what I ask myself is, what new information can I generate so people like Con can implement things that help people? So that's the approach I've taken for this talk. And I want to deal with some very basic concepts that I think speak to the magnitude of this problem, and really what are the mechanisms of this disease and the biggest predictors of this disease. So we'll get started with the most boring stuff. So a few conflicts of interest. A biotech company is taking form here at McMaster that I'm part of. It's related to this work. It does not directly have an impact on what I'm going to tell you. And I do hold patents in this area that are at various levels of approval. Again, they don't directly relate to anything I'm going to show you. So really, what are the objectives today? And what I wanted to tell you, and I hope you can walk away with, is an understanding of insulin resistance during this state of prediabetes. And as Constantine elegantly put it, the state of insulin resistance, and how inflammation can plug into this process. And we're just starting to really appreciate this now. And the other thing I hope you walk away with is an understanding of how the immune system can talk to the metabolic system and why that's important. And this is in the context of insulin resistance. And I'd started to put this talk together, and I was going to tell you about all the immune cells involved and all the cytokines and all these great mediators that we have lots of information about. But what I really wanted to get to is, how is this triggered and why is this even important? Where is this coming from? So I took all that out, and I'm just going to focus on one potential variable. And I will preface this with, this is a lot of my interpretation and my opinion, and I just want to get that out right away at the start. So if we think about our paradigm, what we're trying to solve here, the obesity, immunity, diabetes connection. So the simple fact of the matter is, after Dr. Simon presented this obesity epidemic that's facing us, is that we eat more. We take more calories in, okay? And this is US data. Canada's not that different. And what I'm showing here is the number of calories that are taken in by a person in a single day. So I think that is the critical variable here. This is across decades, but what has actually happened since the 60s or 70s is that the number of calories per day has been increasing, and it's been increasing a lot. It might not look that impressive if you think, well, there's maybe 600 extra calories, but remember, that's every single day across the population. And 600 extra calories equals a lot of fat mass when you do the math. So part and parcel with this, it's really only two sides of this equation: what you take in and what you burn. So, and we're burning less. Okay, and what this is showing here is simply that it's a measure of the calories that you expend at work across this time frame between the 60s and now, every day, and it's going down. So, and there's really no refuting this, that we're taking in more calories and we're burning less. Okay, so I am a big fan of the first law of thermodynamics, and I really, my wife can tell you this, I really do get upset when we forget this in the popular media, and we forget it all the time. There's always a magic solution to obesity. And if you believe in the first law of thermodynamics, if you're taking in more calories and burning less, you're going to be gaining weight. So really, I think this says it all. I really like this cartoon because it is the first law of thermodynamics. Nobody talks about it, right? Especially in the popular media and even in the research field, that if you're going to study obesity, you either believe in this or you don't, okay? If there's an excess of calories, they're going to be stored. So we will get more scientific. But so why is this important? Okay. Well, if we think of this in relatively recent evolutionary terms, we're headed for a dangerous position here. So it's true, we're healthier than ever if you look at this evolutionarily. We live longer, we live better lives, but we're at a strange impasse where there's some evidence emerging that if we continue to go as we are, we're actually going to decrease our quality of life across the population, across the globe, and decrease our lifespan, or our life expectancy. So we're at a really interesting tipping point. And this idea that metabolic syndrome, and one of these factors in this syndrome being diabetes, playing a role in the disease burden is something that we're obviously interested in studying. So if we look back a little further in evolution, again, we're trying to connect obesity, immunity, and diabetes. And here we're going to connect obesity, the metabolic component, and the immune system. Well, it's not surprising if we look at what's required for life. And there's a few definitions of this. But my opinion, and those of several others, is you need to replicate or reproduce, you need to metabolize, and you need to adapt. Okay? And it's not surprising that two of these, the metabolism or growth and the adaptation that can take a form of pathogen defense, are highly linked. Okay? And this goes a long way back in evolution. Okay? And so here are some thoughts from some colleagues. So if we think back 600 million years and we look at the fruit fly, we had one organ that's responsible for a large part of immunity and for fat metabolism, and it's the fat body in Drosophila. So this has then emerged from this. Evolution has given us adipose tissue. That's used to store fat in the fruit fly in that tissue. And the liver, the hepatocytes, also important in immunity and detoxification. And the great cells in here have just emerged as an entire system that's responsible for protecting us. Okay, so it's really not that surprising that immunity and metabolism are heavily linked. Okay. So I think Dr. Simon made this perfectly clear, and it was an elegant discussion about how obesity is the biggest predictor of diabetes, and that obesity is associated with increased inflammation. And this is by no means, diabetes is not an island in this relationship. There really are a multitude of diseases that are linked with increased or changed inflammation responses. Okay? So diabetes does not stand alone here. So really, what are we trying to get to as a metabolic readout here? So if you think of diabetes, Dr. Simon went through this, that as an increase in your blood glucose. So we need to talk about two factors here: glucose and the major hormone that controls it, insulin. So really, insulin can only do this in two ways. You can have more insulin, or it can work better. And when things go wrong, you can have less insulin, or it doesn't work as well. So what happens in obesity? So this is I like to keep things simple, that if you eat and it has carbohydrate in it, your blood glucose goes up. The job of insulin is to drop that blood glucose. It's also very important, as Dr. Simon said, in the regulation of fasting blood glucose and production of glucose from the liver. But so the question really is quite simple. Does obesity affect insulin secretion, or does it affect insulin action? And so there's now a wealth of information that initially, and as Dr. Simon pointed out, the therapeutic window here is that obesity impairs insulin action, and we call this insulin resistance. And why we're so interested in this therapeutic window is that it's reversible. Once you actually develop overt type 2 diabetes, any therapy will have to target the insulin-producing cells, which is proving very difficult. So can we reverse this insulin resistance? And in order to do that, we need to understand how it's occurring. So I include this slide on almost every talk I give. Usually it makes me hungry, and it is 5 p.m. So we eat a meal, it has carbohydrate in it, our blood glucose goes up. It's the job of the pancreas to secrete insulin. Okay, it senses that glucose, and insulin is increased in our blood. It does two things. It increases glucose disposal, and this is largely in the skeletal muscle and tissues like the fat, but it also shuts down glucose production that's always going on. Okay? And if you think about this, it's quite important that we have a source of glucose in our bodies. We can't be constantly walking around with a bottle of Coke, right? That didn't happen 600 million years ago. We didn't have that. So we have the liver that can release glucose into the blood to fuel our brain and our nerves. They need glucose. So it's the job of insulin to shut that down. It's the job of insulin to take this glucose we just ate and dispose of it so we don't have high blood glucose. So really, when you do too much of this, the calories have to go somewhere, right? If you do believe in this first law of thermodynamics, they're going to go somewhere. And where do they go? I'm showing you here lipid deposition in the liver. So the lipid stained red. So the lipid gets deposited in these insulin-sensitive tissues. And that lipid goes to muscle and goes to fat, of course. The idea here is that fat really has a finite capability to store all this excess calories we're eating, and it ends up in all of these insulin-sensitive tissues. And the net result is insulin can't do its job. Can't shut down production. It can't dispose of the glucose from the blood. Really, there's enough insulin. There's more than enough insulin. The problem is that it can't do its job. And then eventually, there are theories on why this happens, whether it's beta cell exhaustion because they're secreting so much insulin, or processes going on in the pancreas. Insulin can't be produced anymore. That's when you have overt type 2 diabetes. So the question is, there's excess calories, there's excess lipid in these tissues. How does that link to impaired insulin action so we can prevent this last step from happening? So as I think both presenters are saying here, it's not a problem of insulin secretion at the onset. It's actually a post-receptor defect. So insulin can bind to the receptor just fine. There's enough receptors. It's really not during obesity, it's generally not a mutation in the insulin receptor. It's a downstream target. And one of the targets that the insulin receptor requires is called insulin receptor substrate. Big surprise, 1 and 2. And it is required to propagate the signal. So the details here aren't really important. But really, what I'm showing you is after this receptor is activated, if you have aberrant phosphorylation effects, so you have a phosphorylation of this protein that stops its action, it cannot propagate the signal and allow glucose to enter the cell. And as it works out in the liver, it's almost an identical system to shut down glucose production. Okay? It's just that you're not causing transport of GLUT4. So the big question is, okay, how does this happen? What is the central dogma and why this IRS1 protein or this adapter protein is malfunctional? So really, part and parcel with obesity is a rise in lipids, and I showed you these lipids get deposited in these tissues. And one of the dogmas in the field right now is that these lipids can actually activate stress kinases in the cell, which can catalyze this reaction. And as it turns out, the activation of these stress kinases were known for a very long time to be a hallmark of inflammation. During bacterial infection, viral infection, these stress kinases are activated to defend the cell. So then it becomes a question of where to target. So it's been extraordinarily difficult to target obesity. If you just look at the evidence, we're not doing a good job. Okay? It continues to rise. There's a lot of work going on in trying to manipulate that pathway to improve insulin sensitivity or reduce resistance. And now that we know inflammation is involved, there's a group of people that are quite interested in looking at the obesity, inflammation, and insulin resistance connection. So that's what I want to tell you about. So I think we've gotten this far in 2 talks and we haven't talked about what inflammation is. So, and I have a very broad definition of inflammation. It's really just an attempt to resolve harmful stimuli. Okay? I could tell you about all the cytokines and cells involved, but really that's what it is. And so this is not always successful. Okay? And mounting this attempt can actually be fatal. Your own immune system can cause death if it's overactivated. And we're now appreciating, or for some time, that this can promote dysfunction and disease, whether that's in a pulsatile way, an acute event like sepsis, or whether it's a chronic disease, such as associated with obesity. So, as I said, I was going to tell you all about how inflammation manifests, but I think it's a more important question to ask, what's the trigger? Why is there inflammation during obesity? Okay? And so for me, it comes down to there's 3 possibilities. This is a bit of my opinion. So it could be the food itself. These nutrients themselves, such as saturated fat or glucose, could activate immune receptors. Okay? That's not unrealistic given that this tight connection between immunity and metabolism. Second, it could be a self-harm detection possibility. So if there's an energy excess, which is the case in obesity, our own cells could be attacking themselves, such as, and we do know this occurs, death of fat cells that become too big. So the third one is that obesity promotes entry of a harmful agent. So, and what I want to tell you is that bacteria is positioned to be one of these agents, okay? And our gut has a lot of bugs in it that are positioned to be one of these agents. So I'm always interested when I tell people that the gut is involved in insulin resistance, and they kind of look at me funny, and I say, it's not that surprising that the gut's involved in obesity, right? All the food does go through there. So what's in our gut? It's not just the food we eat. There's a lot of bugs in our gut. And so we call this the microbiota. Generally, it can weigh up to 2 kilograms. So a good way to cut some weight is to take antibiotics. It'll only work for a very short time, and I don't recommend it. So this microbiome is quite difficult to handle. We think the human genome is actually quite simple compared to the 3 million genes that reside in our gut. Okay, and some people find surprising that what we actually are, there's 10 times more bacterial cells on you than human cells that you call yourself. So some of the questions we have in the lab are what factors from the microbiota are linked to changes in metabolism, and really, does this represent a potential trigger for inflammation during obesity? So our body spends a lot of effort trying to keep these bugs out. Okay, we don't want them in. And this is the inside of the lumen of the gut here, and we have mucus layers that help protect us. We mount, this would be the inside of the body here, we mount a plethora of responses to keep these bugs out. The fact of the matter is they still make it in. And so here's a bug making it through the epithelial barrier. And they're generally dealt with cells that reside in the area of the gut. And this is all quite complex. But for me, it's quite simple, because if you think about the gut, the next tissue that is right there is the fat. So if you look at the mesenteric gut, it's surrounded, it's surrounded in mesenteric lymph nodes and the fat. So this is positioned to be a relationship to explain inflammation in the adipose tissue. And so as it turns out, there's been some recent evidence that this can occur. So during a high lipid diet or during genetic models of obesity, we've come to appreciate this idea of metabolic endotoxedemia. And I by no means discovered this, but we do work in this area. And the general course of events here is that you have some sort of stress, whether it's genetic obesity, nutritional stress. We're very interested in bacterial stress. And it changes those bugs that reside in your gut. It's called a dysbiosis. And this, at least in the context of obesity, can alter gut barrier function. So once you have an altered gut barrier function, things can get in. So bugs can get in, and if you take this all the way down the series of logic, it's possible that more nutrients could get in. So that's coming back to our idea of there has to be more things getting in the body to accumulate these calories. So what we're interested in right now is what types of bugs get through this system, where they go, can they go beyond that immediate tissue that's right there, the fat, and how can this link to this low-grade inflammation that we see during obesity, and that links to insulin resistance. So this is really the area we're working in now. So I think I need to tell you how important bugs are for glucose homeostasis. So Dr. Simon told us very nicely about how important it is to control glucose. And I can show you one experiment that can manipulate glucose simply by changing the bugs. So it's got a little bit of a yuck factor. Okay, this was fecal transplant, and this was in humans. Okay, now this isn't in mice. And I can show you that it'll actually improve insulin action. So basically, a lab did this in humans. They infused into the small intestine lean bugs into obese people, or obese bugs into obese people. And as it turns out, this is a marker of insulin sensitivity largely in the muscle. So the higher this line here, the line in between the bars, the more sensitive you are. And acutely, in fusing these bugs into an obese person that were derived from a lean person, improve the ability of insulin to dispose of glucose. So this is actually arguing in the absence of obesity being a factor controlling this, because it was an acute experiment. But I think it elegantly shows how important these bugs are. So they can acutely regulate the ability of insulin to dispose of glucose. So what do we do in the lab? We generally use mice. And I want to show you two pieces of evidence. I should show you something from my lab. That manipulating the microbiota can change insulin action or regulation of blood glucose. And what I'm just going to show you is one of the most important measures, which is, can blood glucose change? And it can. All we have to do is, so these are obese mice that we give a high-fat diet, and they have hyperglycemia. And if we put them on antibiotics, it depends what type and it depends how long, but we can reduce glucose circulating in the blood. And this is actually turns out to be independent of any change in energy intake, energy expenditure, and obesity. So we're actually trying to get at how we can manipulate dysglycemia without affecting obesity itself. So we can do the flip experiment. It's quite a bit harder, but we have models of dysbiosis where these bugs are obesity prone, and we can feed them to mice and actually promote hyperglycemia. Now you do have to start with a germ-free mouse, and you do have to be very careful on how you colonize it. They're actually quite tricky experiments to get 2 bars to show you. But if you give them a normal flora, they can regulate their blood glucose better than if you give them this dysbiosis model. Okay, and as it turns out, when you do this flip experiment, it was directly related to an increase in fat mass, and that's the fat mass around the gut that I'm showing you. So as opposed to the experiment where we gave antibiotics, this is showing that colonizing mice with a dysbiotic environment promotes obesity. So we have this relationship between bacteria and fat, and it drives changes through obesity and inflammation, leading to insulin resistance. And we have all kinds of interactions here, okay? And what I haven't shown you is that simply eating a lot of lipid or a lot of fat and obesity itself changes the composition of the bacteria, okay, in your gut. But what we're more interested in is how they get through, because we think that's an important inflammatory trigger. So we're dealing in this area of linking obesity and insulin resistance through inflammation. And I've said it's been largely ineffective to try and target obesity per se, but I think the pendulum may start to be swinging, at least at the research level, in looking at this problem. So if we take a step back, can we look at actually changing obesity using bacterial strategies? So what we can do is we can reconstitute mice with human feces. This is someone else's data. And we can show that increased adiposity across generations is transmissible. So we can feed the mice a high-fat diet, their fat mass goes up. And then all you have to do is take the bugs from those obese mice, it actually represents the human flora, because that's what we were using, and then transmit them to another generation of mice, they get obese. Okay, they increase their fat mass. So they were never on a high-fat diet, and they actually don't eat more. So it's a very interesting experiment. As I said, this isn't ours, but we have similar studies on the go. So if you believe me that I said that this is all under the control of the immune system, that the immune system is there to protect us and to guard against us and to keep homeostasis of these bugs in our gut and elsewhere, so there must be a role here for immunity, and that's where therapeutic targets could be derived from. So one problem we are attacking is how these bugs sense the bacterial cell wall. This keeps the whole bacteria together. We have immune receptors that detect a compound called peptidoglycan. It holds the bacteria together, and of course we've evolved a system to say that shouldn't be here. We're going to fight that. So we did some very simple initial experiments, and there are different types of peptidoglycan. It's all very complex, so we just said, let's just delete this. Let's see if we delete that, what happens. So we can delete these proteins called NOD1 and NOD2, and they detect peptidoglycan. And so we've also gotten this far and not shown you a glucose tolerance test. So this is the regulation of glucose when you have a glucose load. So right away you can see these are all obese mice. They all have been put on a high-fat diet. And in green we have the mice that lack peptidoglycan sensing. They can't sense this part of bacteria. And they're protected from hyperglycemia. And when you give them a load of glucose, the lower the line here and the faster it drops, the more sensitive you are. So they can deal with that glucose and get rid of it quite quickly. Okay? And you can get a lot fancier than this, and you can find out how the whole system is working. But this is what's really important. Simply lacking these receptors can improve your glucose homeostasis. And this is part and parcel with reduced inflammation. We can measure this through a plethora of cytokines or intracellular mediators, but these mice that lack peptidoglycan sensing have reduced inflammation, and they actually have a striking reduction in liver fat accumulation when these mice are on a high-fat diet. So I think what I want to tell you as a take-home message, if you want to connect obesity, immunity, and diabetes, this is the relationship I would propose. We need to consider the bugs that reside in us and how they interact with obesity through inflammation and how that can drive changes in insulin resistance. So with that, I'll say thank you. Members of my lab that are probably, hopefully still in the lab, and a great team at McMaster. Okay, we're actually some of the only people that can do these germ-free experiments in Canada right now. And my Toronto collaborators and collaborators abroad, and the funding that allows this to occur. Thank you.